All right. For those of you that don't know, I'm David Weinstein, Dawson James Investment Banking. It's my pleasure to introduce Jerry Jabbour from Matinas BioPharma. When you think about this company, think about oral delivery, something that you can't normally take orally, that you have to have an IV. This company is paving the way for oral delivery systems where you can change that protocol. Jerry?
Thanks, Dave, very much, and good afternoon, everyone, and a special good afternoon to everyone joining on the webcast. We're thrilled to be back here at the Dawson James 8th Annual Small Cap Conference, Growth Conference. It's been a while since we've been here, but the timing could not be better, and this could not be a better platform for us after the week that we've had. I will be making forward-looking statements as part of my presentation, so I would encourage everyone to do their diligence. But before we dive into the presentation, I just want to say this has been a big week for Matinas. You know, in the last 24 hours, we've doubled our market cap, and it is setting the stage for as big a fourth quarter as we could have hoped for because of the catalysts and milestones that we have lined up. Yesterday, we had the good fortune to be able to talk about a patient who our lead drug provided a complete clinical resolution and cure for that patient from a deadly fungal disease. And so from the close of trading on Tuesday until today, yesterday, we traded 360 million shares. And we traded again today, 35 million shares. At one point yesterday, we were up 500% as a company. And I'm not here to talk about share price, but what I am here to talk about is now we have eyeballs. And if yesterday spoke to anything, it spoke to the potential, not only for Matinas, for the thousands of shareholders who have been loyal and stuck with us for a long period of time. The setup is done. And the fourth quarter we have now lined up where we're prepared to deliver what we think can be transformational milestones and catalysts. Because as Dave said, we are all about transforming molecules and making them orally available. And that fundamentally changes therapies for patients. It unlocks values of some drugs that have been around for 50 years that could never be used the way they are now. And everything we do is facilitated by our LNC technology. It's lipid nanocrystals. It's a unique way that we can encapsulate molecules, get them through the GI into the lymphatics, and gain access to cells in a way that doesn't cause cytotoxicity, where there's no immunogenicity, and allows us to give patients options they've never had before. Our flagship is MAT2203. It was the star of the show at ID Week. We had a pipeline presentation this morning at 8 a.m. Dr. Makovits delivered to a standing room only audience. That is where we're taking the highly potent but highly toxic IV-only drug amphotericin and have now made it oral and safe. And I'll go into the details and the data behind that drug, which provides clinical validation for our LNC technology. But we're just getting started because we're taking that platform and now we're applying it to nucleic acids, to small oligonucleotides. And if you know anything about RNAi therapy, about antisense oligonucleotides, about siRNA, they hold great promise, but they can't be delivered orally and they can't really target things outside the liver. With our lipid nanocrystals, we're talking oral extrahepatic delivery, which could fundamentally change the treatment paradigm for a variety of targets. And here we're focused on inflammation and cancer, and we'll explain why our lipid nanocrystals target those sorts of cells. Our pipeline is full, but we're going to focus on the top two lines here because MAT2203 is the value foundation. It is today the therapy that is saving patients' lives. And right behind it is our internal antisense oligonucleotide programs, where we will have no less than 4 studies reading out between now and the end of the year that can fundamentally change our company. Everything we do is based upon this technology. It is simple, it is elegant, it is self-assembling. It is the combination of phosphatidylserine and calcium. We can deliver all sorts of things in this. It's not truly plug and play, but what we've learned and what we've done with amphotericin is directly applicable to what we now want to do with small oligonucleotides. As I said, we're getting beyond the liver. And that is really the holy grail for anybody that's working with nucleic acids. How do we get therapies outside the liver and directly target cells and tissues? That's what our technology does. And we add the benefit of oral delivery. So you don't have patients in hospitals. You don't have patients who have to get IV. They can actually take this outside of the hospital. And there's a huge health economic component to that. And these are safe, these are stable. And we'll talk about the toxicity, the lack of a real toxicity profile. Phosphatidylserine is the key. Every cell in the body has phosphatidylserine. Typically, it sits on the inner layer of the cell membrane. But when you have a cell that is activated, stressed, injured, a tumor cell, phosphatidylserine moves to the outer leaflet and acts as an eat me signal. And it directly binds with our phosphatidylserine-based lipid nanocrystals. And so unlike lipid nanoparticles, which use positively charged lipids to bore holes into cell membranes and destroy endosomes to get into the cytoplasm, we have an invitation, and we can do it via phagocytosis or fusion with the actual cell membrane. And we are the only ones working with phosphatidylserine and calcium. I'll talk about our patent portfolio, but we have the worldwide exclusive rights to a portfolio of patents that come out of Rutgers and to 30 of our own patents, which we have either filed or issued in the last 5 years alone. We're different from other delivery technologies. Liposomes is an old technology. It provided some improvement over free drug in the body, but it's like a liquid bubble. You can't take it orally. They leak. You get no targeting. Lipid nanoparticles are the rage. For any of you who were vaccinated against the COVID-19 virus, lipid nanoparticles were what was used to deliver the messenger RNA vaccines. Again, can only be delivered parenterally and don't have that targeting feature. And they're toxic. There's a reason why you can't have multiple administrations with lipid nanoparticles because of what they do to cells. With our LNCs, this is essentially a next generation lipid nanoparticle where we get into cells without cytotoxicity, where we can get oral bioavailability, and most importantly, we can go to areas of the body that other delivery technologies can't. And that's highlighted by our ability to cross the blood-brain barrier with our antifungal drug, MAT2203. This is our lead drug. There's no question we are living in an unprecedented time for the threat of fungal infections. And yet we are taking a 50-year-old drug, which is the gold standard. And it is the drug that all physicians would reach for if it didn't kill your kidneys. So much so that after 10 or 14 days, you get irreversible nephrotoxicity. Well, when we applied our technology to amphotericin B, we had patients on it at the NIH for 3 years with no toxicity. All right. So we are talking about fundamentally changing the way the most broad spectrum antifungal drug can be used. And so we are preserving the innate characteristics of amphotericin B, applying our unique delivery technology, and now giving patients and physicians a weapon that they only dreamed of in the past. Our phase 2 hit it out of the park. In cryptococcal meningitis, a deadly fungal infection housed in the brain, you have the double challenge of not only treating that infection, but having to cross the blood-brain barrier to do it. We achieved 98% survival after 2 weeks in these very vulnerable patients. You saw no breakthrough infections. You saw an achievement of sterility with our drug, meaning an absence of infection. And it was safe and well tolerated for 6 weeks of treatment in these patients. So again, this was completed and actually just written up in the journal Clinical Infectious Disease. It was an editor's choice and a major article. And this has become really the sounding board and the reason why we are now receiving compassionate use cases, requests for compassionate use in our expanded access, really from all over the United States. And in just the last 12 months, we've treated infections all over the body. Bone infections, bladder infections, brain infections, lung infections. Our technology takes amphotericin directly to the site of infection and treats it effectively without the toxicity of amphotericin B. Yesterday's announcement was all about a 61-year-old gentleman who had heart disease, lung disease, kidney disease, and had a Candida krusei infection. After just 2 weeks, that infection was gone. And so that was just the latest patient. We had a 14-year-old girl with leukemia that we announced the data at ECMID in April, where we cured her. She had 2 different fungal infections. And so this provides a lot of confidence. These are patients who have no other option. The gentleman yesterday couldn't take an azole or an echinocandin, 2 other types of antifungal drugs, because they couldn't achieve blood levels in urine. So they had to turn to our drug because IV amphotericin wasn't an option. It was too toxic for a patient that had kidney disease. He took our drug and it resolved it. And these are exactly the patients we want to study in phase 3. This is the reason why FDA is going to allow us to do a single phase 3 study instead of the normal 2 phase 3 studies you need to do to get a drug approved. That's a major advantage. This is a drug that has qualified infectious disease product designation, orphan drug designation, and we're targeting our phase 3 study in invasive aspergillosis in patients who can't take an azole, can't tolerate an azole, where there's resistance or drug-to-drug interactions. This is a restricted indication that we think will position us for what's called LPAD, the limited population pathway for antifungal and antibacterial drugs. It allows you to do streamlined clinical trials. And so unlike before, where we were thinking about this drug as a first-line unrestricted indication, that would have required 800 patients and 6 years to enroll a trial. Here, even though our patient population is smaller, this is an orphan patient population, this is a gateway indication, and you're talking 200 patients and under 22 months to enroll this trial. Fundamentally different. And we're meeting with FDA within this month to finalize this protocol. There are already companies who have taken this approach and have been approved by FDA to do these 200 patient studies. So that precedent is there. The discussions we had with FDA in the past, and they're very supportive of this drug. They know the importance of oral amphotericin, but they couldn't get around their own precedent in terms of limiting the number of patients or the non-inferiority margin necessary for a first-line unrestricted indication. Despite this being a small patient population, pricing here could be significant because these are patients with no options. The infectious disease doctor remains in control of the prescription, and we've pressure tested this commercially with the large payers in terms of premium pricing. This narrow focus combined with the FDA clarity is why partners are very excited about this drug now. And we expect now with the FDA minutes over the next 30 days that we're going to be able to move this into a phase 3 study, with a partner in the first half of 2024. Our regulatory strategy is clear, it's plain. This is also a drug that's set up for 12 years of exclusivity on approval. So that's regardless of your patents, you have 12 years of exclusivity, which is a great runway to maximize. And as I mentioned, this is a gateway indication. Because amphotericin has been approved before, this is available under a 505(b)(2) pathway once we establish the pharmacodynamic bridge in this first phase 3 trial, meaning you can expand the label to the treatment of mucor, to the treatment of candidiasis, to the treatment of endemic mycoses, and yes, even potentially to prophylaxis or prevention of fungal infections, primarily in immunocompromised patients, by doing studies in the tens of patients, not the hundreds of patients. So this is a pipeline in a product opportunity, and the study design is pretty simple. The value proposition is real. It's our value foundation, but it's just setting the stage for where we're going next. And this fall, in the next, I would say 45 to 60 days, this is where we're in position now to generate data that could fundamentally change, we think, change the industry and certainly the delivery of oligonucleotides. We've done this before. We did it with remdesivir. We took Gilead's remdesivir. We made it oral. They determined to develop their own prodrug instead, presumably because it was cheaper for them. But we showed that we can take other molecules and do this. The lead up to this was all figuring out which cells take up our LNCs the best. And this is a complicated slide, but it comes down to the fact that macrophage and innate immune cells take up our LNCs at a very, very high rate. They, along with tumor cells, and that is why we're headed towards inflammation and cancer. Macrophage are the key cell for many inflammatory diseases. And the fact that we get such high uptake there increases your likelihood of success in having a therapeutic impact. We've already done in vitro work with chemo agents. We've taken docetaxel, Taxol, a very well-known chemotherapy agent that's highly, highly toxic. We already look better than they do in vitro, and we can make it oral and eliminate that toxicity. But this is just a proof of principle with docetaxel for where we want to take small oligonucleotides in the treatment of cancer. Because our LNCs are the same, whether you put docetaxel in it or you put an siRNA in it, uptake will be the same. But we want to show that we're doing it with a small molecule first, show that we can do it better than the legacy agent. And we have 2 cancer in vivo studies. One has already started, and the second with an siRNA we'll have data on before the end of the year. We're going to do it in inflammation too. And we're talking about IL-17. We're talking about things like TNF-alpha. So we are taking small oligonucleotides, proprietary small oligonucleotides. We are putting them into our technology, and we're going to show that we can orally deliver them and knock down these cytokines. There you're talking about diseases like psoriasis. You're talking about diseases like colitis. You're talking about diseases like RA. Think about an oral Humira. Big opportunities here. And these studies, we will have in vivo data in hand in the fourth quarter, some in the next few weeks. And so this is a very exciting time. As I mentioned earlier, there's so many eyeballs on our stock. We're going to continue to deliver with amphotericin B. We are going to get key answers from FDA. We are going to advance our partnership discussions, but we are going to transform medicine with the first successful oral delivery of an oligonucleotide that has a therapeutic effect. So the LNC platform is set up for us to develop not only our internal pipeline, but think about what Alnylam, what Ionis, what other companies who are working in this space would do if they had the ability to deliver their molecules orally. So this is going to allow us, yes, to drive our own products forward, but an in vivo demonstration of oral delivery of a small oligo, even though it's just an animal study, is a big time proof for pharmas that want to deliver their oligonucleotides orally. And so it may be a little while before we have actual product candidates with small oligonucleotides that advance to the IND stage, but these in vivo data will position us very well, we think, for discussions that are already ongoing to advance. This is a well-protected platform. As I said earlier, we have the worldwide exclusive right from Rutgers, but we also prosecute all the IP. So everything since 2015 that we have filed is Matinas IP. And we have gotten 20 issued patents over the last 5 years. In the last year alone, we filed 6 patents. This isn't cookie cutter, because everything you put into the platform requires a different strategy, new IP, something that's not obvious. So this is something that we think will have legs for a long time. We have a great team. We have a great board. Herb Conrad was the former US president of Roche. He was the chairman of Pharmaset when he sold that company to Gilead for $11 billion. He's my 91-year-old former chairman that calls me every day and kicks my ass. But he's a great leader and ceded the chairmanship to Eric Endy, a medical doctor, former Merrill Lynch lead analyst. Carl Icahn thought so much of him, he put him on the board of Biogen and Genzyme to shake those companies up. And so we have sort of the right jockeys. And with the LNC platform, we think we have a thoroughbred. And the fourth quarter is chock full of milestones. Yesterday wasn't even a milestone that we had listed on this chart. Of course, every opportunity we have to change a patient's life is meaningful. But from a data perspective, what we have upcoming here, even after our FDA meeting, we're going to show biological activity with these oral oligonucleotides, not 1, but 2. Then we're going to be in position to share, we think, disease therapeutic effect, disease data with these small oligos, again, in the fourth quarter. We think the clarity around 2203, we're batting 1,000 in the treatment of patients in our expanded access program. That only increases the confidence that partners have that this is a drug they need to put in their bag. And if we are successful in doing what we've already shown in vitro and in small in vivo studies in delivering oligonucleotides orally, we will be in position for new platform collaborations that are supported by data. Our bio and tech collaboration was about a dream that didn't work on the first try. And BioNTech's going to be sad that they walked away, because now our in vivo studies, we believe, are going to show the ability to deliver nucleic acids orally. We have a solid value foundation. We have clinical validation of our platform, but huge upside in the new areas of science. I'll stop there and see if there's any questions in the room. But again, thrilled to be at Dawson James. Thank you for the platform. And I've been with this, I founded this company. I've been in the CEO seat since 2018. I've never been more excited for a 2-month period than I am now. The team is energized. We have wind in our sails. And given the activity of the last 2 days, we have a lot of eyeballs on us. And so we're going to supply the fuel, and we're looking forward to big things in the fourth quarter. Thank you. Any questions? Yes. Sure. So we have cash until next September. We have 217 million shares outstanding, all common. We have no warrants. We have no debt. Burn rate is about $5 million a quarter. The last time we sold any stock was at $1.93 off our ATM in January of 2021. Our last raise was with Piper Jaffray in 2020 at $1.55. And we didn't sell any stock yesterday.
Out of all the stuff you have in your pipeline, what do you think the largest revenue producer is going to be?
So if we're talking about actual tangible drugs, MAT2203 is a drug that, if it becomes that pipeline in a product that you're envisioning, that's a drug that annual sales in the US alone will be over $700 million a year, US alone. And the utilization outside the US is likely 1.5 times that. Yes, sir. Yeah, so you're not getting composition of matter on phosphatidylserine and calcium, right? So it is formulation patents which take into account the molecule that's included. Now, if you were to put in a molecule that also had its own patent protection, that would provide dual protection in that situation. Thank you very much. Have a great rest of the conference.